All right. Everybody. Can you guys hear me okay? Can I go through the speaker? I'm going to have a weird tail for this thing, okay? I couldn't get me a wireless one, so let's see if I don't trip myself. You guys can take that. Since I'm going to trip on this wire or not. Let's see. A little bit higher. Okay, so you guys got rained out this week, huh? Don't have to fear, you're going out next week. Woo-hoo! Okay, so I am Professor Drew Lamar. I am the official and unofficial math geek in the biology department. There are other ones that are math geeks, but I'm the most outwardly so. My background actually is in mathematics. I got a PhD in mathematics, and I'm in a biology department. So let that be known to you that whenever you're thinking about your career track, it may not at all be what you expect. Okay. So one of the things that I wanted to do for you guys today is to talk to you about the many opportunities that there are at William & Mary for quantitative biology. Sometimes you may have heard it called mathematical biology, but I like quantitative because it makes it more broad than mathematical. And I think if I said, for many of you, if I say mathematical, it's like you're narcoleptic. You're like, so quantitative hopefully is not quite the bludgeoning of a word that causes fear and whatever else in your body. So here we go. This was there was an article put out a while ago in the Ideation magazine, which is a magazine about research and scholarship at William & Mary, highlighting biomath on campus. So here are a bunch of people who are a lot of faculty in different departments who do biomath. Okay, you have people in biology, obviously. You have people in mathematics, obviously. But you also have people in applied science. You've got people in chemistry. You've got people in physics. You've got people, all over the place. Maybe even in history. I don't know, actually. But so some of the people that you'll see here in psychology. This is Professor Joshua Burke. This is Gregory in psychology. This is Gregory Smith in applied science. They're part of the neuroscience program. How many of you are neuroscience majors or want to be neuroscience majors? So this was a project that they were doing in neuroscience. This was an undergraduate research student, Kia Hardcastle, who worked with both of them in different departments. Okay. Here we have, on the cover, is Junping Shi from the math department. This is Leah Shaw from the applied science department. And this is Ron Lipschitz from VIMS. How many of you know of VIMS? Virginia Institute for Marine Science. It's sort of a sister institution to William & Mary. They're very close in Gloucester Point area. And they do a lot of marine science stuff. Okay, so then there's quite a bit of mathematics and modeling going on over there. Okay? So what they're looking at right here are a bunch of oysters. So they did a project with looking at oyster shell regrowth and how to get oysters to be much more prolific in the Chesapeake Bay because they've been somewhat decimated. Okay, and over here, here I am looking much more groomed. Okay, shaved head, no beard. This is me, Drew Lamar. This is Sarah Day in the math department. She's actually my wife. That doesn't mean anything at all. And this is our ex-student, I guess, alumni, Allison Korish, who is now working as a quant, actually in New York City. But her project was looking at the synchronization of fireflies and looking at the mathematics behind how some fireflies are known to just pulse together. It's pretty cool. There's actually a species in North America that does this, over in the Rocky Mountains, I think. Something like that. It's in the States. But the one that everyone knows about, there's a species in Thailand as well. They have this making this synchronous bursting. It's pretty neat. This was in the article. They want, of course, whenever you see an article like this, you think, okay, this is really just flawed. We do not write equations on glass. It drives me crazy, okay? I would probably fall down and get dizzy because my eyes would be focusing in and out, right? So, but anyways, they wanted a picture of Kia, this is Kia again, putting math equations on glass in the building, McLaughlin Street Hall. I have no idea if these equations are even related to research. Just a funny note here. I went to my first year of grad school at UCLA, and I took a complex analysis, fancy name for a class. And the professor, because this is in LA, right? This professor actually had worked for Hollywood many times for movies, and one of his claims to fame was he wrote equations on the board in an episode of Baywatch. I'm not sure you guys are too young to know about Baywatch, but okay, it's good stuff. David Hasselhoff. No idea. Anyways, so or like Good Will Hunting, you got equations on the board, right? You know math, you can look at that and be like, that's not at all what they're talking about. It's pretty fun. Okay, so I just wanted to show you this link here. This is where it's going to get fun. Here we go. There's actually a link on campus for biomath. Here it is. Very fancy. And so you can take a look. There's a lot to see. There's faculty, there's postdoc, there's a journal club every Friday at noon where we read journal articles and get together and talk about it. There's a lot of courses here, so you can click in here and see a lot of the different types of biomath courses that are offered around campus. Many of them are offered in biology. So it's good to take a look for those that are interested. Okay. Hot off the press. How many of you, let me poll you now, how many of you are not afraid of math or like math or find it interesting? Raise your hand. High and proud, come on. Okay. How many of you don't care? Okay, that's good. How many of you have a hatred for math and quantitative? Okay. Good, I'm like, good. So, how many of you, let's keep asking again, how many of you are thinking, I want to be a biology major because I hear that there's not much math in it? Yeah? Okay. That's, I'm sad to say you're misinformed. It's becoming more and more quantitative, both the field of biology as well as the major itself over time. So, if you want to have no quantitation, you better get out of here quick. You can graduate in 2 years, it will be less quantitative. Okay, here's the thing. Is it new CAMS mathematical biology major and minor that I want to mention to you guys? CAMS stands for Computational and Applied Math and Statistics. Okay. It's really interdisciplinary. Okay, and so because of that, it doesn't live in a department. It lives in the Charles Center. In the interdisciplinary program. And for the minor, when we designed it, we really had the biology major in mind. Okay, so those of you that like quantitative ideas, that are interested in like building those skills, this minor was designed for you. Okay, and it stresses 3 major components or skills of quantitative literacy. Here's the first one: mathematical modeling, whatever that means. Okay. Statistics and data analysis. And computer programming, scripting. Simulation, essentially. Okay, these 3 skills are really great and important skills to learn for your biology major. Okay? In this minor, what this will do is it'll allow you to go a little bit deeper into those areas, those skills. Okay, quantitative biology courses at William & Mary. This is just a quick sample. There's one that I teach that's going on right now called Introduction to Quantitative Biology. Okay? This actually counts for your math requirements in biology. Okay? It's not officially on the web pages yet, but it does count. It will be official soon. There's also a course, a sister course in the math department called Introduction to Mathematical Biology. They use a scary math word. They're similar, but this course, the one that I teach, stresses all 3 of these things. Okay? And I think this course might stress mainly this one. Introduction to Biostats. Okay, that's offered in the spring. Usually offered in the spring. Okay, it will be offered in the spring this coming year. I will be teaching that as well. Okay. And some other ones. This is applied science in biology, random walks in biology. If you want to learn about stochastic effects and stochastic things and noise and variability in biology. Besides biostats, this is the course for you. And then the neuroscience majors may have heard of this course. Cellular biophysics and modeling. If you're a neuroscience major, raise your hand if you've heard of this course. Keep your hand up if you're afraid of this course. Okay. You may have heard word of mouth from other students who have taken it. That it's a challenging course. And again, but it's, I think, really important, especially if you're a neuroscience major, which is, very, very quantitative science. Okay. Why quantitative biology? He looks so defeated. Don't ask me why I had this up there. It's sort of a no context type picture. I think when I used it before in a class, I just had the question why, and so my argument was, well, it's all about sexual reproduction, right? We're biologists. That's why we're here, right? So that's why I had it, but I figured, just keep it up because it's funny to watch. I just love how defeated he looks. What's up? Oh. Okay, so let's talk a little bit about why quantitative biology, and then I'm going to start with experimentation. You guys are doing labs. In labs, you're doing experiments, observational or experiments. This is very true, okay? And I put this slide up there. I'm not sure if you guys can read it from the back. Some people think scientists exclaim, Eureka, when doing experiments. But they're way more likely to say, and these are a bunch of things that I probably shouldn't say or I'll get fired. Okay. And then the last one says, I hate science. Okay. I put this up for a couple of reasons. Many of you may not really know what it's like to do experiments and research, okay? These labs that we have for you, we prepared for you in your undergraduate's introductory biology course, are not your standard run-of-the-mill labs. This is not something where you're going to do the same kind of thing we've done before, that everyone's done before previously, that, it's canned protocols. It's not going to be that. These are research-based labs. Okay? You guys are doing and participating in research. As such, when you do research, things go wrong a lot. Okay? So that's what this is trying to tell you, is that when you're doing experiments and doing research, it's just going to be a lot of this kind of stuff. Not that depressing. It can be very fun whenever you have these things happen. There's not an explosion or fire. There's not someone on fire running away around or something like that. So that's good. But it's just to point out to you that you have an opportunity, a great opportunity of doing a research labs. Okay? So be prepared for things to possibly not go right. And don't freak out. Okay? I promise don't freak out. Don't freak out. All right, let's talk about it. Why mathematical modeling? So here's the real world. All janky and corners and crazy. Right? I showed this to my daughter, and she thought it was a mouse. I tried to make it something that didn't look like anything. She's like, oh, look at a mouse, how cute. Oh, whatever. There you go. The real world is a mouse. And when you're thinking about the scientific method, right, this is what you start with: a hypothesis or a prediction. Okay? And where do you go from there? Well, you have experimental design, and this experimental design is probably one of the most important steps, okay, of the scientific method. And that leads to setting up an experiment, or either observational or in the lab type experiment. Okay, so what lab or field work? You do measurements on your experiment, and you get data. And a lot of times, once you get to this step, you're like, now what? Well, that's where statistics comes in. Analysis. You take that data, and what do you do with it? Well, how do you take that data and use that data as evidence for or against your hypothesis or prediction? Okay, so you have your conclusions, you either reject your hypothesis or not. Suppose you screwed up somewhere along the way. If your experimental design was messed up or whatever, or something bad happened in your experiment, and your data was just garbage, you can redesign, go back, and do it again. You can spend some time looping around here for a while. Things go well, maybe what will happen is that you can extend. You get results, you get your conclusion, that'll lead to more questions, more hypotheses, you go back, wash, rinse, repeat. Okay? Is this all looking familiar? Yeah? But the question is, how does all this really relate to this? Okay? So here we are playing a game of scientific method, and the question is, this arrow here, how do we know if what we're doing here is any indication of what's actually going on? All right? And that's one of the ways where statistics can help. Okay, but besides just stats, there's mathematical modeling. Okay, let me explain that. So here's reality. That was our maze. Here's our hypothesis prediction, same as before. Except now we have two branches. On this side is the one I just talked about, experimental design. But on this side is a analogous thing, which is model design. And this is where you can use it in a mathematical model. So the key thing here is you can think of a mathematical model as an experiment. Okay? And more importantly, one of the reasons why you might want to do a mathematical modeling experiment, let me ask you guys this. Why might you want to do this as opposed to this? Any ideas?
You don't waste time and resources doing something you need to know it.
That's right. So time and resources, okay? So let's say you're trying to ask a question about something that happens to, something biological that has generation times that are really long. You may not have the time to just stick around to actually run the experiment. You may not have the money to do it. That's a great one. What's another one that's very timely? I'll give you an example. Ebola. Yeah.
You can't do natural field experiments with Ebola because that involves people getting Ebola.
Right, so there's two things here. Number one, if you're doing an observational experiment where you're going out there and there's already an epidemic and it's occurring and you're just collecting data, there's the safety issue. Right? People are doing that now. In fact, there's lots of interesting articles out there on the quantitative analysis people are doing to try to figure out who's patient zero and all this kind of contact tracing, different statistical reasons. But also, if you think about epidemiology in general, do you think it's a good idea to just take humans and infect them with something and see what happens?
Probably not.
Probably not, right? You lose your job really quickly. So that's not a good idea, okay? So that's another area where mathematical modeling can come into play. Right? If you want to be morals, if you want to have morals, then you can do this. Maybe not style, but morals. Okay. Maybe you're not cool because you're doing math, but whatever. Okay, so over here, remember we did measurements on our experiment and we got data. Over here, you can do simulation or analysis and you get results. Okay, fantastic. So these are kind of similar. And then with those results, or with that data, you can do, again, same thing. You use statistics or analysis on the results and the data to go to your conclusion, reject or not. Right. Same thing here. You can do a redesign if you screwed up or you needed to change something. Maybe you set up your experiment and you got data and it didn't really answer the question you thought it was going to answer. Same thing with the model. Suppose you got the results that didn't look quite right. Right. You look at the results and you say, okay, I know that this species does not behave this way. This is bogus. Something's wrong. Refine your model. Change your assumptions. Run it again. Okay. So you can get dizzy spending a lot of time here. And then if things go well, and you can get more questions, you go back and you extend the model or you revisit your hypothesis. Does that make sense? A couple other things. This step is really nice. A lot of times the golden something, I don't know what the term's called. What you're really shooting for when you're doing a mathematical model is you want to be able to do the modeling alongside of the experiments. Okay, because what that allows for is validation. Look at your model results and look at the data that you collected and you say, do they match? Okay? And that's a good way to validate your model. Okay? That's that big red arrow that says, is this actually making sense with the real world or not? Keep in mind, every time you do one of these step downs, you have a possibility of increasing error. Okay? So by the time you're down here, you're so far away from the reality that, you're just essentially playing around. Okay? All right. Some great questions. We'll keep that on that. Do you think we can know reality for certain?
No.
No.
It's always theory, right? Not just proof.
Okay. So this is okay. It is theory, right. So you have theory of gravity, evolutionary theory. So even in physics you have theories, right? Mechanics, classical mechanics. Is classical mechanics the truth? No, why not?
Relativity.
Relativity. But is classical mechanics accurate? Yeah, sure. It depends on your scale, right? But if you're talking about the scale of like this room, right? Then classical mechanics is really accurate. It's very, very accurate. Very successfully accurate. But it's not quote unquote truth because of relativity, because of quantum theory, right? All these different things that get added on later and make you realize, oh yeah, so it's not quite the truth. So even in physics, all right, now, I'm not saying that theories are, well, it's just a theory, right? That's what everyone says, it's just a theory, right? We like to have debates with people about evolution being just a theory. I think this is actually may have been mentioned in your class. Theory. What are theories? Scientific theories. You want to know? No one knows what scientific theory is?
Explanation or observation supported by evidence.
So observation supported by evidence. Okay, so that could be. There's actually different tiers. Where's the theory lie in that? Because yeah, it's definitely a hypothesis or observation that's supported by evidence. Right? But where does it lie on the spectrum of very little evidence to lots and lots of evidence?
It's kind of confirmed.
So it's
Extremely high, right? So the theory is like, hypothesis or that observation has lots of evidence backing it up. And when I say lots, I mean like lots. Lots of independent replication, lots of independent scientists out there trying to disprove it and not being able to. Okay? And that's what scientists do, that they try to disprove. We're a skeptical bunch. And we want you to be skeptical. Okay? But we want you to be rationally skeptical. Okay? It's healthy to be skeptical. But the question here is certainty. There is no certainty. There's only evidence. Okay? So it's nothing certain or known. What is evidence? We throw this word around like we know what it is, but what is it? How do we know whenever you have something in front of you that it's evidence for something? It's actually a big philosophical question. We're getting into like the philosophy of science here. But what is evidence? What is statistics? Finally, getting into statistics. That's statistics. Statistics looks at the question of certainty and uncertainty, and it quantifies it. Okay? So if you wanted to have a phrase that rolled off your tongue when someone said, what is statistics? This is a fantastic one to have, for you to have like right there to grab. Statistics is about quantifying uncertainty. I've measured data, I have it in front of me, I want to know how certain I am of this data that matches the real world. That's statistics. It's quantification of that question. That's why I love teaching this class, because you can't not have these philosophical science questions about what is evidence without, it's a part of the whole course. All right, so here's some other little tidbits. Statistics is a technology that describes and measures aspects of nature from samples. Okay? Basic idea is you have this thing, this massive biological thing you're trying to probe and ask questions about. You cannot and will not very rarely be able to measure everything that you care about, right? If you ask a question about all the species, like a species of Carolina wren, you're not going to be able to measure every single Carolina wren in the world. Right? So what do? You sample. You maybe grab 20 of them. And then you measure those, and then you get something, and you say, okay, well, how do I know if this thing that I've measured relates to all the Carolina wrens out there? How close is this estimate that I have, how is that going to relate back to the whole population? Okay. Statistics. Okay. That's where it comes from. Again, it lets us quantify the uncertainty of those measures. It makes it possible to determine the likely magnitude of measurements' departure from the truth. It's about estimation. This is one of those skills that, as an undergraduate biology major, you do well to learn about estimation skills. Okay? It's the process of inferring an unknown quantity of a target population using sample data. Target population, all Carolina wrens. Sample, 20 Carolina wrens. What you're measuring, how does it relate to the whole? So many of you have probably heard that probability and statistics are paired together. Right? The class of probability and statistics. Does anyone hear that? Yeah. You guys wouldn't mind if I killed it, would you? Biologists. Don't do it! It's okay. I'll save any kind of bug in my house except for roaches. Can't stand them. Alright, anyways, so here's what statistics is. Okay, you ready for this? Alright, all Carolina wrens up there, populate, that's called the population. Your sample is your 20 down here, 20 samples of Carolina wrens. Okay? Probability has to do with up here. It's usually a probability model, a mathematical model. Okay? And then statistics, and what that does is that says, assuming all these things about the upper population, what do I expect to see if I do sample? That's probability. Statistics goes the other way. I grab the sample, I measure things, I want to know how does this sample relate back to the population. That's statistics. Okay. So probability is theoretical. It gives you predictions about what you expect to see if you sample. Samples go the other way. They infer, statistical inference, what's going on in the population from a sample. That make sense? That's a really good nugget to know. I had this flipped, I think. This should be probability and that should be statistics. I think. Is that right? Are you guys writing this down? I gave the slides up. I'll fix it, and then when they post the slides, it'll be fixed. Scout's on. Scout. Easy. Okay. Let's do a little bit of fun. Okay, great, whatever. Professor Lamar. But really, why would I need it? In the real world. When am I ever going to use this stupid equation? I hate math. I never use a quadratic equation. Okay, there's a momentum shift that's happening in mathematical education. And the shift is the following. What is the pinnacle of math for a student? Considered to be usually. What's the last course you would take as a mathematician and you go, yeah, no. Calculus. That is a little bit outdated. When do you use calculus in the real world? See, thank you. Never. Not never. But. How about statistics? All the time. Data analysis and stats. Okay, so this is what the shit that's happening is that a lot of people are saying, you guys should not be having your pinnacle be calculus, it should be statistics and data analysis. Because that's what you see in your everyday life. And the problem is that people will use statistics and data analysis to convince you of the truth. Right? So it gets used as a weapon more often than you realize. And so I wanted to show you this quickly. Headlines from a mathematically literate world. This is a great website. Not sure if you guys can see this. So, for example, our world, market rebounds after assurances from Fed chair. And the literate world says, market rebounds without clear causal explanation. Okay. There's some other really good ones. Gas prices hit record high, unadjusted for inflation. Gas prices hit record high in a vacuous, meaningless sense. Baby food sales going up over time. Babies are getting hungrier. Okay. Let's see. After switch in standardized tests, scores drop. After switch in standardized tests, scores no longer directly comparable. Oh, here we go. Controversial program would cost $50 million in taxpayer money. Controversial program would cost 0.001% of taxpayer money. Okay. So, numeracy, quantitative literacy, statistics, data analysis, this stuff gets used as propaganda. And if you are not equipped, besides just being horribly skeptical and cynical, which is not very healthy over the long term, then you will be ripe for them to influence you. Okay. You should know this stuff so that you can bludgeon other people with your beliefs, right? Isn't that why we're here? Yeah. Okay. Okay, I'm a big Redditor. Another reason to know stats is that it shows up a lot in conversation. Oh, on Reddit. This was an example where they were trying to, they thought that there was a downvote brigade, which essentially is a bunch of people that go out and try to downvote certain things. They agree to downvote things. So they needed evidence for this. So what do? This whole thread of conversation says things like, We need to do a t-test, guys. 95% confidence interval. Someone do it. Look how excited he is. Stats! And then let's do this right, because it's experimental design, right? Let's do this right. Find other subreddits that link to things and run an ANOVA. Hey, you guys heard that before. I wanted to see if SRS is any different from other subs. So you have categories of subs, and you want to know if the means are different. That's ANOVA. The sample must be randomized for this to work. Experimental design. And then this guy says, considering I just did matched pairs tests for data analysis, I actually could, but I just finished my assignment today, and I don't really feel like doing it again. Ha ha. Another one. This was a paper published recently. Why most published research findings are false. Isn't that nice? Of course, yeah, it's a little bit meta, right? Because this is also a research paper. Right? So is it false? Oh. All right, what this is getting at is the problem of replication in science. A lot of times, if a lab does an experiment, it will cost a ton of money and a lot of resources, and it's almost nearly impossible for someone else to replicate. And the environment does not look favorably upon a lab replicating findings. Everybody wants to have new findings. Okay, there's a big push that don't replicate. You're not going to get tenure. You need to do your own stuff. Okay, and what's happening is that you're getting lots and lots of results. Very little replication, so you're not getting the building of evidence. Okay. Add on to the fact that when you have data and you're doing statistics, you're never certain, ever. Okay, you always just have evidence. So there's always some percentage that what you've seen is actually just a fluke. Okay, it's a small probability chance of it occurring. This is natural variability, and so that's why you have replication. Key important stats idea. Here's another one. Someone put out a paper not too long ago saying that ESP, they proved ESP is a real thing. Okay? This gets back at the 95% confidence idea. Again, if you don't replicate, if there's some small chance that you'll see a pattern that will look like this ESP is real, right? And you do one experiment and you see that pattern, then you might say, ha, ESP is real. But if you do it another 19 times, it'll come up as not real. Right? So this again gets back to replication. It says, yet the episode has inflamed one of the longest running debates in science. For decades, some statisticians have argued that the standard technique used to analyze data in much of social science and medicine overstates many study findings, often by a lot. So this is getting at, again, the uncertainty in statistics itself. As a result, these experts say the literature is littered with positive findings that do not pan out, effective therapies that are no better than a placebo, slight biases that do not affect behavior, and brain imaging correlations that are meaningless. This is littered with statistical terms. Okay? It's important that you know them, okay? And you will learn them in this class throughout the semester. In particular in this lab, because you'll be doing some of these tests at the very end. Okay? So to quickly tie back to what you're doing in this lab, the next 2 weeks you're going to collect data, and then the last week you're going to do the analysis, and that's when you're going to do a lot of these statistical techniques, which I'll talk about. Anybody remember this? Nate Silver? Raise your hand if you've heard of Nate Silver. Back in the elections, right? 2012. What did Nate Silver do? Anybody want to tell me what he did? Good.
You got every single race correct except for the North Dakota Senate race.
Yeah. He predicted all the races except for one.
Predicted Florida correctly in the morning of the election.
Yeah.
So it was the closest state to the entire race.
Yeah. So it's pretty amazing. This is to show you the power of data analysis and statistics for prediction. Okay. The title's great too. It made statistics sexy again. As far as I'm concerned, I never went out of style. Okay. In the remaining 15 minutes, I want to tell you the two main ideas of statistics. Okay? These are good nuggets again. Number one is estimating parameters. Okay? As I mentioned before, all the Carolina winds, some sample. You're trying to measure something. What is that something that you care about? Okay? What are the parameters, the numerical parameters of a population that you care about? So here's just a quick picture again of what population versus sample means. Here's a population of red and white beans in a whole statistic lab. have a cup full of red and white beans and do something. Right? This is your population. Suppose you have 80% white, 20% red. You sample that 100 beans out of that 3,000, and you, rarely is this ever going to happen, have 80 white and 20 red. Okay? But this is all you're going to see. If you do an experiment, most of the time, if you do an experiment or observational experiment, you're only going to see this, and you're going to have to try and infer, does this proportion of white to red somehow, how does that relate back to the whole population? How certain am I that this is the true proportion of white to red in the bigger population? Okay. Does that make sense? You bored yet? Thank you for being honest. Yes? All right. Well, I'll bore you some more. How about that? Deal with it. Okay. Types of data and variable. This is really key, okay, when you're talking about stats. What is a variable? It's any characteristic or measurement that differs from individual to individual. Phenotype, height, weight, speed, reproductive rate, etc. These are things that you might care about measuring in a population. Many of them are numerical, some of them are more categorical. We'll talk about that. Well, categorical. So one type of variable is categorical, where it's membership in a category or group. So, for example, you could have gender. Genotype. Okay? Those would be categoricals. There's two different types. There's a nominal one, no order of inherent categories. It doesn't really make sense how you order them. And then ordinal. Inherent categories have an ordering. And before you say that this is why math is boring, because it has all these definitions, does biology have any terms you have to memorize? Are you kidding me? Right? So. Okay, numerical. Quantitative measurements. All right. Continuous. Any real number in a range of values. Okay, this could be like decimals. Discrete. You don't have to write it down. First of all, remember, the slides are going up. Indivisible units of measurement. The counts. These are you do a lot of counting of things. Okay? So that's what a discrete is. And data is any raw measurement of one or more variables made on a sample of individuals. Okay? It's like so obvious that you're like, oh God, move on. But these are important because it is really, really important that you know what is the data before you even try to do the analysis. Okay? Because what stats you do on that depends critically on what your data is. Okay? So, boring maybe, important yes. Okay, so here's an example of one way to visualize with a histogram. This is a continuous variable, and we use this histogram. All right, temperature is the vertical axis. I'm going to tell you some descriptive statistics. The mean. Everyone knows the mean. Right? Woo-hoo. I'm sure anybody. How many of you calculate your own grade with the mean? How many of you don't calculate your own grade with the mean? Okay, how many just are like, I don't want to even answer your question. Fair enough. Okay, so the mean measures what they call central tendency. It's sort of the, what they call the average is another word for it. There are other measures of central tendency. The median is one of them. But in statistics, you'll learn more about all the different types of things, ways of measuring. Where's the middle? The typical. Variance. That's the formula. That's pretty. Okay. So you talked about this in class with ANOVA, right? ANOVA is all about variances up the wazoo. Okay? Lots of different types of variances. So you talked about it in class, so I'm not going to spend much time on it. But again, it gives you some idea of dispersion or spread around the mean. Right? How spread out is your data? And you're looking at reference to the mean. Standard deviation is dispersive variance. Okay? And 66% of measurements are approximately within one standard deviation from the mean. Okay? So it gives you some indication about how much and where of your data is located around your mean. This is only precise if you have what type of distribution? Normal. You guys got to speak up. Seriously. As you get older, you don't care anymore, so you just kind of blah. So it's good. Okay, here's the second type of thing. So the first was, I'm measuring something, I want to know how certain I am on that measurement. Right? I just want to know, the average height of a sample and how does that relate to the population and how accurate do I think I am. Okay. The next thing is hypothesis testing. Okay. So I have time to go over one example, and I shall do that, and then I'll go over the other examples right before you do your data analysis for this take thing. Okay. So here's the example I'll go over right now. Contingency test between categorical variables. Suppose you have two categorical variables, and contingency is a fancy word, but it'll make sense in a second. What is it good for? Absolutely nothing.
Say it again now.
Whoa. Wake up. So it's to test if there is an association between two categorical variables, right? Are they associated? Right? Do they depend on one another? That's the idea. Okay? Does this depend on that? So here's your example. Can parasitism alter behavior of the host? What's the answer? Yes. Hard, right? Because remember, we don't know with certainty. We have evidence for it. In these situations,
Yes.
the answer is going to be yes. There's evidence for it. And if, in this situation, it'll be that way. So the setup. Trematodes of the species use 3 hosts during their life cycle. Okay? So this is fun. By the way, parasitism is fascinating area of study. So you'll see that here. So first you have the shorebird, who likes to poop in the water, and horned snails get a hold of that. Right? Parasites are in the poop, and this is an uninfected one, and that's when they open one up. This is an infected one. I think this is I think this is all the parasites in here. All the white you see here is all parasites. And then what happens with the horned snail is that the killfish eats the horned snail. And what's awesome about that is that where does this parasite go? To the brain. Zombie fish. Fantastic. And then what happens with these fish? They get eaten by the bird. And then the cycle continues. Okay? Here's a video of fish in a tank, and I want you to spot the one that's infected. So, do you think that's going to increase the chance for that fish to be eaten or not? Yeah. And so what's interesting is that not only does the fish do that, it tends to be near the surface sometimes too.
So it like makes the fish suicidal.
Yeah, it's just they go crazy. Right? It's like too much coffee or something. All right, so here's the data they collected. Observed frequencies of fish eaten or not eaten by birds according to treatment to infection level. So here's your 2 categories. The fish that were eaten by birds and the fish that were not eaten by birds. Other category. Were they uninfected, lightly infected, or highly infected? And then you have counts. Nice little table with counts. Sometimes you'll see a mosaic plot of this. So here's relative frequency. Not eaten versus eaten. Red is not eaten, yellow is eaten. And then here's your infection status. Tell me, by looking at that, do you think that being eaten depends on your infection? Yeah. Okay, but we cannot just say, hey, look, it looks like it. It must be true. We have to quantify that certainty or uncertainty. Okay? So what we do is we have our observed frequencies, and then we create our expected frequencies under a null hypothesis. And you mentioned null hypothesis in class as well. The null hypothesis is that there's no association between those two. And under that assumption, in the probability world, you can write down what you would expect to see in your table under the assumption of no association. So this is what you would expect to see under the null hypothesis. And then that's what you actually see. So you can see they're pretty different numbers, right? So now you got to compare the two. So what you do is you use something called a test statistic that compares the observed versus the null hypothesis expected. And what you're seeing here is distance from expected is the numerator, and the denominator makes it relative. Okay? So you can see this is kind of a distance, kind of like ANOVA, the variances and stuff. If this is large, what does that mean? You would reject the null. So it's got to be large enough. I'll say air quotes and stuff. How big does this need to be to reject the hypothesis of no association? Well, this is where p-values come in. the probability of obtaining the data are more extreme, assuming the null hypothesis of no association is true. So under the null hypothesis, assuming it's true, it's the probability of obtaining that data that you actually see. If that probability is small, there's no way I would have seen this given the null hypothesis. It's very, very unlikely. Then you can reject that. You have evidence against the null. So then you use significance levels, which is also mentioned in class. And what does everyone always use? 5%. But remember, this is not like a hard cutoff. Probability threshold at which we would reject the hypothesis of no association in our p-value is smaller than this level. But again, how big does it need to be? Well, there's also something called the critical value. This is the threshold value of the statistic at which we would reject the hypothesis of no association if that statistic is larger than this value. And this depends on your significance level. Okay. The smaller your significance level, the larger your critical value. It's got this inverse relationship. That's what we calculated for this. The critical is 5.99. Is that larger than that? Yes, so we would say that there is a significant association, or there's evidence for a significant association. And then you can actually calculate the p-value directly if you want to. Approximately 10 to the minus 15. Is that small? Yeah. Okay. So there's evidence, guys. Not truth, not yes, evidence that parasitism does affect behavior in this system. Okay. All right, I think you guys faded like 10 minutes ago. Yeah, or 30 minutes ago. So get out of here. Have fun in the woods.